So let's just review the concept of the endomembrane system. All proteins that are destined for any part of the endomembrane system, remember, are going to be synthesized by ribosomes on the rough ER. They will either be injected into the ER if they are going to be an internal type protein or a secreted protein, or they will be injected into the actual membrane itself if they are going to be a glycoprotein, a transmembrane protein, etc. Once these proteins are produced in the ER, they will travel to the Golgi apparatus where they will receive some sort of modification, and there is where they will mature. At the trans side of the Golgi apparatus, they are going to be sorted out and then be delivered in vesicles to their final destination. This slide here reviews the components and the trafficking within the endomembrane system. Remember, we have the nuclear envelope that extends into the rough and smooth ER. Any protein that is going to be secreted or part of the plasma membrane or part of a lysosome or any other part of the endomembrane system is going to be made by the rough ER. It's going to be packaged in vesicles delivered to the cis side of the Golgi. It will then be matured, glycosylated if necessary, and then it will exit the Golgi through the trans side. From there, it will be delivered to a lysosome if that's its final destination, if it's a hydrolytic enzyme, or it could be delivered to the plasma membrane. It will either be secreted if it is an internal protein, or it will be inserted into the plasma membrane if it is an embedded protein. We will continue this lecture by discussing other membrane-bound organelles that are present in the cell. Keep in mind that these are membrane-bound, meaning that they are surrounded by a lipid bilayer. However, these organelles are not considered to be part of the endomembrane system. Two of these important organelles have a role in converting energy. One of these organelles is a mitochondria. This is responsible for harvesting energy from sugars and fats, so this is the site of cellular respiration. All eukaryotic cells will have mitochondria present in them. Chloroplasts are specialized organelles that help to harvest solar energy and convert it into chemical energy. This is the site of photosynthesis. However, these are not present in all eukaryotic cells. They are only present in plant cells and some algal cells. Both of these organelles are considered to be semi-autonomous. They have some of their own DNA. They even have some of their own ribosomes that will help them translate this DNA into proteins. By being semi-autonomous, this means they can replicate independently of the rest of the cell. This is important when we consider where these structures originated from. The endosymbiotic theory is a theory that is proposed to explain the evolution of the mitochondria and the chloroplasts within eukaryotic cells. It is originally believed that these organelles were considered to be their own bacteria that were ingested by a prehistoric eukaryotic cell. Some of the evidence that is there to support this idea is the fact that they have their own DNA and genes that are outside of the genome of the eukaryotic cell. They can carry out their own gene expression and their own protein synthesis. If we look at the structure of the DNA of chloroplasts, we can find that it is very similar in sequence to the blue-green algae DNA. There are also many similarities of these structures to various types of bacteria. Both bacteria and these organelles can divide on their own when needed. If we look at the overall structure of these components as well, we can see that the inner membrane of these organelles is very similar to a bacterial membrane. Mitochondria even have bacterial cell walls. If we also take a look at the ribosome structure of these organelles, it strongly resembles bacterial ribosomes rather than the eukaryotic ribosomes that are present in the cytosol. This slide here depicts the endosymbiotic theory. In the top of this image, we have the ancestral eukaryotic cell, which we will term the host cell. So as the host cell ingests the bacteria, which would be the mitochondrion, we can see that a symbiotic relationship would exist. When a symbiotic relationship exists, that means that two parties that are involved in that relationship would benefit in some way. In this instance, you would have the host cell benefiting from the ability to manufacture ATP as an energy source, and the mitochondrion would benefit by having a protected environment to reproduce in and to survive in. Because of this symbiotic relationship, remember your theory of natural selection. This would give that host cell a distinct advantage over others in the population. This new host cell with a mitochondria present would have an increase in fitness, meaning it would be more likely to survive and reproduce in that environment. As that cell was splitting, it would also pass on mitochondria onto its offspring. As a result, we would see this evolving in the population as a whole. Similarly, when we have the ingestion of the chloroplast, now we would have a eukaryotic ancestral cell that now has the ability to utilize solar energy as an energy source. This would once again give those cell types a selective advantage over others. We will now take a look at the structure and function of the mitochondria. The mitochondria is responsible for producing the bulk of the ATP of the cell. ATP is considered to be the cellular currency of energy for eukaryotic cells. The ATP is going to be synthesized by the mitochondria by breaking down sugars and fats. It will use the energy in these molecules to help produce ATP. Cells that are considered to be more metabolically active, like muscle cells for instance, will have many more mitochondria present in them. This is because they have higher energetic demands, and so they would need more mitochondria to meet the ATP demands of the cell. The structure of the mitochondria has a double membrane. So that means that it will have an outer membrane which is smooth and resembles more of a eukaryotic type cell and origin, whereas the inner membrane, which resembles a bacterial cell membrane, is going to be highly folded into cristae. Keep in mind a general principle that when you have highly folded areas, this typically is done to increase the surface area to carry out a certain function. So if we consider the structure of the mitochondria, the inner membrane is going to be highly folded because this is a site of ATP production. Because we have two membranes, we will then have two compartments to the mitochondria. The innermost compartment or space is termed the matrix, whereas the compartment or space between the two membranes can be referred to as the inner membrane space. This slide here will help you understand the organization of the mitochondria. If we start with the innermost chamber or space of the mitochondria, we refer to that as the matrix. The matrix is going to have the DNA. It's also going to have the free ribosomes that will help carry out translation. This is going to be surrounded by the inner membrane. The inner membrane is going to be highly folded, and those folds can be referred to as the cristae. Outside of the inner membrane, you have the intermembrane space, which is then enclosed by the outer membrane. We will now take a look at the structure and function of the chloroplast. The chloroplast contains chlorophyll, which is a specialized pigment that will help carry out photosynthesis. The chloroplast is responsible for harvesting solar energy and converting that solar energy into chemical energy, specifically producing sugars. The chloroplast also has a double membrane. However, inside of these membranes, we will have an additional layer of organization. This is what we refer to as the thylakoid. So in the thylakoid is going to also consist of a lipid bilayer. The thylakoid is going to be the internal structure where the site of photosynthesis occurs. Thylakoids are going to stack on top of each other to form the granum, which we can see referred to in the image to the right. These stacks are then going to be enclosed in the stroma. So because we have this additional layer of organization, we will also have an additional compartment or space to the chloroplast that we don't see in the mitochondria. So that means that we have three different compartments to the chloroplast. The first compartment or innermost compartment will be referred to as the thylakoid space. The thylakoid space is going to be surrounded by the thylakoid membrane. This together makes up the thylakoid. The thylakoid is a lima bean-like structure. that is stacked on top of each other. These stacks will help to form the granum. If we see in the image below, please try to orient yourself with respect to the thylakoid. You will see that those thylakoids are stacked together. You can see a cross section of those thylakoids to see the innermost chamber, which would be the thylakoid space. So these stacks of thylakoids are going to be interconnected with membranes. This is where photosynthesis is going to occur. So all of those thylakoid stacks are going to be enclosed in the inner membrane of the chloroplasts. So we have the thylakoid space inside, surrounded by the thylakoid membrane. We will have those thylakoids enclosed in the inner membrane. The space which is enclosed by the inner membrane is going to be called the stroma. Just like we saw in the mitochondria, we will also have an intermembrane space that exists between the inner and outer membrane. So if you refer to the diagram below, we have the inner membrane, which encloses the chloroplasts and will enclose all of the granum. You will then have your intermembrane space, and then all of these structures will be enclosed by the outer membrane. You can also see that there are DNA and ribosomes present within the chloroplast as well. This will help make the chloroplast semi-autonomous, as mentioned before. We will now get to the structures of the cytoskeleton. The cytoskeleton is important to give support, strength, and shape to the cell as a whole. The cytoskeleton can allow for motility and contraction of the cell. It can also help to anchor organelles within the cell, and it can also help anchor cells to each other or to a surface. The cytoskeleton can also be important in regulating many cell functions, such as the cell cycle and cell division. There are three types of cytoskeleton features: the microtubules, microfilaments, and intermediate filaments. It is important to keep in mind what are the base units that make up this structure, how these structures are formed, and their overall function in the cell. The first cytoskeleton feature we will learn about are the microtubules. Microtubules are made up of monomer units, alpha and beta tubulin. Alpha and beta tubulin will join together to form a tubulin dimer. These dimers will then arrange themselves to form a tubule-like structure. With respect to size, microtubules are considered to be the largest of the cytoskeleton features. They have many different functions in the cell. They're very important in helping to separate out the chromosomes during mitosis. Microtubules also are important in moving organelles or vesicles throughout the cytosol. They can also provide the base structure for the flagella and cilia, so they are responsible for helping to move the cell within its medium. Microtubules also help to form the centrosome. The centrosome is a structure that is made up of two centrioles. These help to form the microtubule organizing center, which will grow the microtubules, which then attach to the chromosomes to help separate them. So microtubules are very important in cell division. If we take a look at the diagram to the right, you will see the overall organization of those dimers in forming a tubule. You will also see where the microtubules lay with respect to the cell. You can see that there are many microtubules that surround the nucleus. We can see that in the blue structure in the middle of the cell. Microtubules will also be found throughout the cytosol. This is because they're responsible for moving many of those organelles throughout the cell. Microfilaments are the next cytoskeleton feature that we will discuss. Microfilaments are made up of actin subunits. Actin is a small globular protein that is very similar to alpha and beta tubulin. However, these actin subunits will simply coil around themselves to form a fibrous-like intertwined structure. Microfilaments are very important in changing cell shape, in contracting the cell. They can also be involved in cytoplasmic streaming, meaning that they can move organelles from one location in the cell to another. But they're very important in moving the cell, specifically forming and contracting the pseudopodia. Overall, if we take a look at the relative size, the microfilaments are going to be the smallest of the cytoskeleton feature. If we take a look at where they lay within the cell, we will see microfilaments throughout the cell because they're involved in cytoplasmic streaming. But if you take a look at the image to the right, we will see that there's a high concentration of microfilaments directly below the cell membrane. This is because of their important function in moving the cell and maintaining the cell shape. The last cytoskeleton feature that we will take a look at is the intermediate filaments. Intermediate filaments are made up of fibrous proteins. If you recall, the microtubules and microfilaments are made up of small, round, globular proteins such as tubulin or actin. In this case, for intermediate filaments, we have more fibrous-like proteins such as keratin that comprise these filaments. These fibrous proteins are coil-shaped. When we take a look at the structure of the intermediate filaments, if you imagine that you have a coil like a telephone cord, that would be considered your fibrous protein unit. If you were to take multiple telephone cords and wrap them around each other, this would be considered your intermediate filament. Intermediate filaments are, with respect to size, relative size, they lay in between the microtubules and the microfilaments. So intermediate filaments are smaller than microtubules, but bigger than microfilaments. These intermediate filaments, because of their coil-like structure, they are going to be very resistant to tensile forces. They're going to be responsible for anchoring the nucleus, for forming the nuclear lamina, and for anchoring organelles to each other. So they're very important in maintaining the overall organization of the cell. If we take a look at where they lay within the cell, you can see to the image to the right that there is a very high concentration of intermediate filaments that lay right around the nucleus. We also will have them throughout the cell structure, as you can see in the green filaments throughout the cytosol. Now we're going to take a look at unique structures that can exist on cells, specifically the flagella and the cilia. These structures are going to be comprised of microtubules. These microtubules are going to be arranged in a specific way in space. They have a 9+2 arrangement, which refers to the microtubule arrangement of the structure. There's going to be doublets of microtubules, so 2 microtubule units will be anchored together. This is termed the doublet. In the flagella and the cilia, we have a 9+2 arrangement because there's going to be 2 central single tubules that are going to be in the middle of the structure. Around that structure, we will have 9 doublets that are paired up that will arrange themselves in a circle. If we take a look at the cross-section in the image to the left, you can see that we have the central microtubules in the middle, and if you count, there are going to be 9 doublets encircling that. Both the flagella and cilia are going to be anchored to a basal body. The basal body is also termed a microtubule organizing center. So this is going to be at the base of the structure because those microtubules that actually form the structure originate from that basal body. So as those microtubules grow out from the basal body, the overall structure will grow as well. With respect to their function and structure, cilia are going to be very, very small. They're going to exist in large numbers, and they're typically found on the surface of the cell. Flagella are much larger than cilia, and as a result, a cell will only have 1 or 2 flagellum. In the top right image, we have an example of a flagellum on a sperm cell. On the bottom right image, you can see that we've got a eukaryotic cell that has millions of cilia on the outer surface. We are now going to discuss the cell wall. The cell wall is only present in some eukaryotic cells. It can also be present in some prokaryotic cells as well as some protists. Animal cells, true animal cells, never have a cell wall. The cell wall is a structure that always exists outside of the plasma membrane. In plants, the cell wall is primarily made of cellulose. If you remember, cellulose is a very strong carbohydrate macromolecule that is based off of beta-glucose units. Because you have a beta-glucose linkage, cellulose exists in sheets of fiber By arranging different layers of sheets, we comprise the cell wall, which can provide a lot of support to the cell. The cell wall is going to be responsible for resisting osmotic pressure that is building up in the central vacuole. Together, the central vacuole and the pressure that exists between the central vacuole and the cell wall will help to maintain the rigidity of a plant cell. There can be two cell walls present in a plant cell. The primary cell wall is present in most all plant cells. However, the secondary cell wall is only going to be present in more of your woody type plants. So the secondary cell wall is only present in a few plant cell types. The outermost wall is considered to be thin and flexible, while the inside primary cell wall overall is much stronger. Here we take a look at the overall structure of a plant cell. We've got the central vacuole, we've got the cytosol, the plasma membrane, and that plasma membrane is going to be surrounded by the plant cell wall. In the enlarged image, we can see the presence of the secondary cell wall as well as the primary cell wall. This would be in between the two plant cell types, where we have the middle lamellae that is interconnecting between the two cells. This image here depicts the overall arrangement of the cellulose fibers. By arranging the cellulose into sheets that the fibers run in different directions, you can provide a lot of strength to the cell wall. Now we are going to discuss the extracellular matrix. The extracellular matrix is the substance that you can find outside of the cell. Animals do not have a cell wall, but they do make an extensive extracellular matrix. The extracellular matrix, or ECM, is mostly consisting of glycoproteins that are secreted by the cell. Remember, anything that is secreted by the cell would have to go through the endomembrane system. So these glycoproteins in the ECM would be produced by the rough ER packaged in vesicles going through the endomembrane system to then be secreted by the cell into the ECM. Collagen is a main component of the ECM. Over 50% of total protein in the human body is actually comprised of collagen. Fibronectin is another important protein that is present in the ECM. This helps to attach to integrins on the cell surface. So this helps to anchor the ECM to the plasma membrane. These integrins are also going to be connected to the actin filaments, or the cytoskeleton, that underlies the plasma membrane. This image depicts the extracellular matrix. Note that there is a high percentage of collagen protein in the ECM. You will also have various types of proteoglycan complexes. This is comprised of a core protein that is attached to many different carbohydrates. This is going to be important in hydrating the cell and maintaining a high water concentration so that nutrients and waste can easily dissolve into and out of the cell. This ECM is going to be held in close association to the cell membrane itself. If you take a look, you can see the fibronectin proteins that attach to the collagen fibers. This is then going to anchor into the integrin proteins. The integrin proteins are transmembrane proteins that span the entire plasma membrane. These integrin proteins are then going to be connected to the microfilaments of the cytoskeleton. So overall, you can see the importance of the ECM and its components and how it attaches to the surface of the cell membrane and then helps to anchor the cell with respect to its cytoskeleton. So in the previous slide, we have seen how the cell can anchor itself to the ECM. This is done by integrin proteins. However, the cell also will need to attach itself to adjacent cells. For cell-to-cell connections, we can have various types of junctions that will allow the cells to communicate and also to adhere to each other. Between cells, you will have gap junctions. These are channels that will allow for cells to share the cytoplasm. These are also called communication junctions. These are very important to allow for rapid communication or synchronization of cells. These are going to be very important in any tissue types that have to contract at a similar time, such as your intestinal tissue, your heart tissue, etc. Within and between cells, you will also have tight junctions. Tight junctions will help to form a barrier and to separate out compartments of the cell. You will also have desmosomes. These are points of very strong attachment between cells. This is because they are comprised of intermediate filaments and keratins that will help provide a very strong connection point. If we take a look at the overall organization of these types of junctions, you will notice that the tight junctions are found at the top end of the cell. Underneath the tight junctions, you will have the desmosomes that will strongly attach adjacent cells to each other. And then finally, at the basal or bottom region of the cell, you will then have your gap junctions that allow for communication between cells. Keep in mind that we also have the extracellular matrix that is attached by the integrin proteins. So we will end this lecture by looking at just another depiction of these cell-to-cell attachments. Here you can notice that there are some adherence junctions which are based off of actin filaments that are depicted in this image. However, they are not depicted in the previous image. But adherence junctions are, once again, just a way that cells can attach to each other, but this time using actin filaments. We have our desmosomes that consist of our intermediate filaments, tight junctions that also consist of actin, and then your gap junctions at the basalmost area that provide open communication between cells.